Good afternoon. I'd like to begin by just asking you to turn off your cell phones or put them on vibrate mode if you have them. I'd like to introduce myself. I'm Greg Germino. I'm the Deputy Director of NIDDK. And I'd like to, on behalf of Dr. Griffin Rogers, welcome you to this week's session of the Wednesday afternoon lecture series, both those of you here in the audience and those of you on the web. Metabolomics is an emerging field with tremendous potential for advancing our understanding of human health and disease, and for the development of personalized medical interventions. We are pleased today to have one of the foremost practitioners of this technology to discuss his work. Dr. Newgard uses an interdisciplinary approach involving gene discovery, metabolic engineering, and comprehensive tools of metabolic analysis, such as mass spectrometry-based metabolic profiling and NMR-based metabolic flux analysis, to study the mechanisms of diabetes and obesity. Christopher Newgard is the director of the Sarah W. Stedman Nutrition and Metabolism Center and the W. David and Sarah W. Stedman Distinguished Professor of Pharmacology and Cancer Biology at the Duke University Medical Center. Prior to coming to Duke in 2002, Dr. Newgard was the Gifford G. Touchstone Jr. and Randolph G. Touchstone Distinguished Professor in the Department of Biochemistry, as well as co-director of the Touchstone Center for Diabetes Research at the University of Texas Southwestern Medical Center in Dallas. Dr. Newgard is a prolific scientist, having authored over 200 peer-reviewed and review articles, and he's been the recipient of numerous awards, including the Kayla Grotsky Award for Outstanding Basic Science Research from the Juvenile Diabetes Research Foundation in 1999, the Kayla Grotsky Award for Outstanding Basic Science Research from the Juvenile That's repeating myself. The Outstanding Scientific Achievement Award from Lilly from the American Diabetes Association in 2001, a Merit Award from NIDDK, also in 2001, the Solomon Burson Prize of the American Physiological Society in 2003, and a Freedom to Discover Award in Metabolic Research from Bristol-Myers Squibb, also in 2006. It is also important to note that he's a terrific public citizen of science, having served on several study sections for the NIH and for NIDDK. At NIDDK, we know him as an outstanding collaborator who is fearless with technology, highly innovative, and a tremendously supportive mentor and advisor for many young investigators. Please join me in welcoming Dr. Newgard to the NIH.
Thanks a lot. Thank you. Wow, that was a terrific introduction. I'd like to meet that guy someday. It gives me great pleasure to be here, be among a number of friends, and to be part of this Wednesday afternoon lecture series, which is obviously very prestigious, and it's a great privilege and an honor. As you heard in the introduction, what I'd like to do is talk to you today about comprehensive metabolic profiling, and in the first couple slides, I'm simply going to describe a platform that we've been building at the Stedman Nutrition and Metabolism Center that I lead at Duke University. But much more importantly, what I want to spend time on today is the application of these tools to chronic conditions and diseases. Most close to my heart would be obesity and type 2 diabetes, but as you will see increasingly also we've been branching into cardiovascular disease, which is obviously of interest to many of you. So let me just go through two slides describing what is our Metabolomics platform. And I do this because metabolomics is a word with no intrinsic meaning in the English language. So I could be, setting up a paddle tennis court for all you know. But the truth of the matter is, and the other thing, of course, is that different people that do metabolomics, some of whom may have come to this audience, will have different views of what metabolomics means. So what do I mean when I come here to speak to you? So we want comprehensive tools, and deeply comprehensive tools for metabolic analysis, and the tools have to be versatile, because we jump back and forth, as many of you do, between cultured cells, animal models, and clinical samples. There are two main instrument platforms for this technology, NMR and mass spectrometry. Some years ago, we chose to focus on mass spec. The major reason for this is that it's more sensitive than NMR, and at the end of the day, you can measure more metabolites at one time with mass spec in a quantitatively rigorous fashion than you can with NMR. But when you look at mass spec, there's more than one way to do mass spec in the context of measuring metabolites in a static kind of way. You can do targeted mass spectrometry or you can do non-targeted. We do both. Let me emphasize that we do both. But much of what I'm going to tell you about today, if not all of what I'm going to tell you about today, employs the targeted mass spec methods. So what do I mean by that? We're talking about simple instruments here, GCMS and tandem MS, and it's targeted because we're adding cocktails of stable isotope labeled standards to every sample and measuring the analytes of interest against those stable isotope labeled internal standards to read out in real physical units, nanomolar and micromolar. And I think this is a really important thing if you want to do clinical epidemiology, is to know what the heck you're measuring and know its real concentration in physical units. And so this is important to us. Now, if you're going to insist on doing it this way, there are going to be limitations. Not every small molecule metabolite in the metabolome, and it's estimated that there are perhaps 6,500 metabolites in the human metabolome. Not everyone is going to have a commercially available stable isotope labeled standard that you can purchase. For that and other reasons, our current capability in aggregate is about 200 metabolites, which seems rather puny compared to the metabolome of 6,500 metabolites. But these metabolites are distributed over 9 modules or classes, which actually do a pretty good job of assessing energy metabolism, the metabolism of the 3 major macronutrients, fats, carbohydrates, proteins, amino acids. And if you just go through these modules for a second, free fatty acids, total fatty acids, long and short chain acyl-CoAs, acyl-carnitines. These first 5 are like a home movie of the metabolism of fatty acids. As you are a free fat, as you become esterified, as you become CoA activated in the cytosol, as you become oxidized and catabolized in the mitochondria. We're reading each of those fates in these modules of 15 to 50 analytes in these targeted analyses. We measure organic acids, the TCA cycle intermediates and related molecules. We measure amino acids and urea cycle intermediates. The newest module is the precursors of purine biosynthesis, as well as many nucleotides, as well as ceramides and sphingolipids. This is a constant work in progress. So we're adding modules currently for sterols, phospholipids, and eicosanoids. And as I said, we also do use non-targeted metabolomics for discovery purposes, not so much for clinical epidemiology, because we think it's very difficult to reproduce. But we do non-targeted in 2 ways. One is shotgun or open-ended GCMS, so you're looking at everything that you can see in the small molecule spectrum with a GCMS instrument, but aided here by a now 900 compound retention time locked spectral library that doesn't solve the quantification issue, but does tell you when you're comparing biology A to biology B, drug addition, no drug addition, gene knockout, no gene knockout. If a peak flies or several peaks are higher in biology A versus biology B in terms of peak area, the retention time locked spectral library can you quickly identify that analyte via its retention time, and then you can turn that into a targeted assay with the appropriate stable isotope labeled standard. So that's a game that we play. We use the broad spectrum tool for profiling, for discovery, and then we always try to convert back to a targeted quantitative assay. And the same with the sort of Rolls-Royce instrument in our shop, which in this case happens to be Agilent's QTOF tandem MS, ultra-high pressure liquid chromatography tandem MS. Now we're talking about being able to resolve at least thousands of peaks, small molecule analytes, but the spectral libraries and the stable isotope libraries have even further to go with regard to this technology than for the simpler instrument GCMS. So we've had lots of interesting experiences in discovery with these tools, including a wonderful collaboration that we're having with Jeff Gordon's group on trying to understand deep sequencing of RNA transcripts coupled to metabolomic profiling. The GCMS is an excellent tool for this because some of the metabolites that you want to look at in the microbiome are not necessarily classical energy metabolites. They're weird sugars and other things that bugs will make. And so I really think that there's tremendous potential for these approaches, but I'm not going to feature that today. I'm going to take you on a more circumscribed journey, the journey of low-hanging fruit, using these targeted approaches in the context of clinical investigation and its translation to mechanistic work. Okay, now, in addition to being able to statically profile large numbers of metabolites in different kinds of samples, if you really want to understand the metabolic status of cells and tissues and organisms, you need to understand flux through pathways in which those metabolites engage. And we've been doing that for many years, as I'm sure many of you have as well, using classical radioisotopic techniques. But in addition, we're very fortunate to have a long-standing collaboration with Dean Sherry, Shawn Burgess, and Craig Malloy at my former institution, UT Southwestern Medical Center. And they use stable isotopes and metabolic flux measurements by NMR via techniques like mass isotopomer analysis. And so we can couple metabolic profiling statically with flux analysis to get a much better understanding of how metabolism is changing in different pathophysiologic or physiologic circumstances. And I'll try to give you an example of that in a moment. Now, once you have all of this instrumentation, it needs to be complemented by a lot of other accoutrements, if you will, informatics, statistical analysis, and modeling tools. Very obvious, you need that. This is high-density data. We have robots in our shop. I never thought I'd see the day, because you really want to maximize throughput and you want to minimize interassay variability. So precision in sample aliquoting is an important piece. We've created a hormone and cytokine multiplex assay core. So not only are we profiling metabolites, but we also want to profile important peptides that influence metabolism, such as, all of these hormones that have come on the scene: ghrelin, leptin, adiponectin, NPY, etc. And then last but not least, we're able to do physiologic profiling, both in animals and in humans, by body composition, glucose tolerance testing, indirect calorimetry. So end of the day, what I've just described for you, and I'm done talking about technology, this is just our flavor of how we're going to do metabolic analysis. And what it really, I hope what it really is a metabolic physiology platform, not just a sort of dry metabolomics metabolic profiling platform. And that's what we've tried to put together. All right, so how do we use it? What do we learn? That's really the important thing here. And so here's what I'm going to try to cover. I'm not going to do the last point, and I'm going to do these other bullets in thirds. So I'm going to start by telling you about metabolic signatures of chronic human diseases that we all care about, diseases and conditions, including obesity, type 2 diabetes, and cardiovascular disease. And it will be no surprise to anybody in this room that people with obesity have different levels of metabolites than people who are lean, or that people with type 2 diabetes have different levels of metabolites than people who don't have diabetes, or people who have cardiovascular disease can be picked out by metabolic signature. But what is exciting and interesting, and what I'm going to try to drive home with two examples today, is that we really are now at a place where metabolomics can be used at baseline to predict disease progression and to predict outcomes of interventions. And I'm going to give you two examples of this that I think are exciting, and I believe it's really only the beginning of the potential of these tools for such purpose. Now, when I work with my very gifted and very energetic clinical research colleagues at Duke and elsewhere, I'm a PhD, I'm not an MD. If you ever see me around patients, call the police. But they are excited and are ready to quit the scene if we're able to come forward and say, here's a biomarker profile of the progression of disease in a patient, or here's a biomarker profile that predicts the outcome of intervention. But we as PhDs, and indeed MD PhD scientists as well, want to do more than that. If we see a metabolic signature of a disease state that we perhaps weren't anticipating, what really excites my academic laboratory, my basic science laboratory these days, is that the metabolic signatures that emanate from these kinds of studies can be used as hypothesis generation engines for mechanistic studies in cells and animal models. Almost a retro-translation, if you will, from the bedside back to the laboratory, back to the bench. So I'm going to start with this, then I'm going to tell you about integrating metabolomics with other omics sciences for identification of novel regulatory pathways, and I'm going to finish by talking about how these tools can define cellular signaling mechanisms, and I'm going to particularly use the example of glucose-stimulated insulin secretion with you today. I won't have time to talk about the very powerful insights that one can gain from metabolomics applied to transgenic mouse models, but we've been experiencing this in the NERSA consortium, the Nuclear Receptor Consortium that Ron Margolis manages so very effectively. And we've been using these metabolomic tools to understand the tissues and the pathways that are influenced when our colleagues like Bert O'Malley and Mitch Lazar and others knock out nuclear receptors or co-regulators with no prior knowledge of what to expect from a metabolic pathway perspective. These tools can shine a light on where's the action in terms of the tissue and where's the action in terms of the specific metabolic pathway. I just won't have time to give you some of the really exciting examples we've had about that today. So let's move to metabolic signatures and how they generate hypotheses that can be tested. So once we built this appliance at Duke about 6 years ago, we were very fortunate to have a very talented clinical investigator join the Stedman Center as our Director of Clinical Research, and that's Dr. Laura Svetky, who is a clinical trialist that is involved in several important multi-center clinical trials, NIH-sponsored, including the weight loss maintenance trial, where Laura is looking for the effects of particular kinds of dietary and behavioral interventions at the level of hypertension and obesity. And so Laura and Lillian Lien and Andrea Hack, some of our young faculty, clinical faculty, went out together in the Raleigh-Durham community and very easily recruited 74 obese subjects with an average BMI of 37. What was more difficult was to recruit 67 lean subjects, but they persevered and managed to do it. And one of the exclusion criteria in this study that we decided on was no diabetes medications, no serious illness. But with a BMI of 37, it won't surprise any of you that these subjects were severely insulin resistant, okay? So what we're really doing here is comparing obese insulin resistant to lean insulin sensitive. And the exercise here, which was really sort of an unknown outcome exercise at the time, was if we take this now more granular, more comprehensive metabolic profiling tool out and compare these two populations, are we going to see things about metabolism of the obese insulin-resistant state that we hadn't anticipated before from the excellent work that's been done over many years where analytes tend to be analyzed one at a time or in small clusters? Is there anything to be gained in knowledge by the broader approach? So we went into the blood and urine of these subjects and performed our targeted mass spec-based profiling along with hormonal profiling and analysis of conventional metabolites by conventional methods. And we put all the data in a hopper and subjected it to a multivariate statistical analysis that many of you are familiar with, principal components analysis. I'm not a statistician, but the way I think of it is principal components tells you which analytes like to cluster together in statistical space. That's the technical description, okay? And it turns out that there were 5 principal components in this data set, clusters of around 8 to 10 metabolites, that together comprised or described about 75% of the variance in the entire data set. Okay, so you're simplifying data into clusters, and then you're finding clusters that have an overall large impact on variance in the data. And several of these principal components or clusters were lipid related. Fatty acids, for example, one cluster was several long chain fatty acids and ketone metabolites. Another cluster was medium chain acyl carnitines. And yet another cluster is the one I show here. The branched chain amino acids valine, leucine, isoleucine, glutamate, C3 and C5 acyl carnitines, and phenylalanine and tyrosine. I had to scratch my head a little bit, but it eventually dawned on me that just like long-chain fatty acids and ketones, or medium-chain acylcarnitines, constitute clusters that seem biologically relevant and significant, this is actually a cluster where you can argue there's biological continuity, not just statistical association. So the 3 branched-chain amino acids are in this cluster, valine, leucine, isoleucine. The first step in branched-chain amino acids catabolism for all 3 branched-chain amino acids is transamination, which usually yields glutamate. So now we can connect glutamate in here. As you go further in the catabolism of the alpha keto acids derived from transamination of branched-chain amino acids, you generate 3-carbon and 5-carbon acyl-CoAs, which read out in our profile as 3-carbon and 5-carbon acylcarnitines. I struggled a little bit more with the aromatic amino acids until I came to realize that branched-chain amino acids and aromatic amino acids are taken up by the same large neutral amino acid carrier. And they actually compete with each other on a class basis for uptake through those carrier proteins that get them into cells. So one idea that we have is that the rise in phenylalanine and tyrosine that accompanies the rise in branched-chain amino acids in the people that we're surveying here may be secondary to that competition for uptake. Okay, but the surprising thing that we found is that when we asked which of these principal components is most strongly associated with insulin resistance, measured crudely here by the HOMA score, it was not fatty acids and fatty acid-related metabolites, but rather it was this branched-chain amino acid-related principal component. And you can see that relationship here, and this was far stronger than any other principal component. All right, let's pause here for a second and ask, have we really learned anything? Because, as I am reminded frequently, 30 years ago, there are a number of laboratories were able to demonstrate that branched-chain amino acids are elevated in obese, insulin-resistant, and diabetic subjects compared to lean. This goes back to Errol Marlis and a number of other investigators as well, George Cahill, Phil Fehlig, and others. So that's not a new observation. But what is new is 2 things. First, it's not just branched-chain amino acids that are up in obese and insulin-resistant subjects. It is also a cadre of metabolites that come downstream in the branched-chain amino acid catabolic pathway. And second, the thing that comes out by having done a broad metabolic profile is that the principal component most strongly associated with insulin resistance is this one, and not lipid-related components. And those we felt were the new features of the finding that we had here. So this is now just an observation, and we have to go further. We have to drive hypotheses, we have to drive validation. So here are the important questions that arose. Do increased branched-chain amino acids and metabolites actually contribute to the development of insulin resistance? Is this simply a biomarker of insulin resistance, or is there actually some kind of cause and effect relationship between these variables? Is it replicable? Is this signature replicable in other human cohorts? You know, as a lifelong rat doctor, I have gained an enormous respect for the importance of replication cohorts in human research, particularly when you're applying omics technologies. You must do this. And thankfully, we've done it in spades, and as you'll see in a second, everything holds up in many examples. And then to take it a step further, is it possible that branched-chain amino acids actually can be predictive of disease or intervention outcomes? So let me answer these questions in a brief way. First of all, with regard to cause and effect, Dr. Ji-an, a longtime senior research scientist in my group, did a 6-arm feeding study in normal rats where she fed the following diets: high-fat diet, high-fat supplemented with branched-chain amino acids, standard chow diet, standard chow plus branched-chain. She had to create a high-fat pair-fed group. And the reason is that when she feeds the high-fat plus branched-chain diet, the animals spontaneously eat less food and attain the standard chow diet weight. And so to be able to say anything about whether branched chains are contributing to insulin resistance, you must feed the high-fat diet at the lower rate of food intake reached spontaneously by the HF plus BCAA group. And another control was to add all of the amino acids isonitrogenous to the supplementation by BCAA. And what we found is that branched-chain amino acid supplementation of the high-fat diet, but interestingly not the standard chow diet, caused a decrease in food intake. But these animals that get the high-fat plus branched-chain diet, which are standard chow weight, are every bit as insulin resistant as the chubby high-fat animals. I'm going to show you that data just to drive this point home in just a second. Insulin resistance is not seen when we pair feed the high-fat diet. So at this stage, what we can conclude, and we have to be very careful in our conclusions, is that in the context of high-fat diet, not in the context of standard chow diet, the addition of branched-chain amino acids to the high-fat diet contributes to the development of insulin resistance in a body weight independent fashion. And I think that's as much of a conclusion as you can make. Now, with regard to mechanism, what we found is that feeding the high-fat plus branched-chain diet caused a chronic activation of the kinase mTOR in muscle and in liver, and this effect was not seen with pair feeding of high-fat or the high-fat pan amino acid diet. So very briefly, just two data slides to support my summary here. First of all, these are the feeding studies, and you can see here's the high-fat diet body weight in yellow, the standard chow in the, what are those, brownish-orange triangles, lower body weight. If you supplement the high-fat diet with branched-chain amino acids, the animals eat less, and now you have animals with a standard chow body weight. But if you look at the glucose tolerance curves here, the high-fat and the high-fat plus branched-chain amino acid groups are equally glucose intolerant, despite the fact that this group has a standard chow body weight. And I'm not showing it here, but if we had done the pair feeding of high-fat to this lower rate of food intake, those animals are normally glucose tolerant. If we further examine the potential role of this chronic activation of mTOR in this phenotype or syndrome, here's what we find. Again, glucose tolerance curves. So you can see standard chow diet here, normal glucose tolerance. High-fat diet causes glucose intolerance. High-fat plus branched-chain, absent any rapamycin treatment, also glucose intolerant. This is a completely different set of animals than the prior ones in reproducing the earlier findings. And then high-fat plus branched-chain with rapamycin given just before the glucose tolerance test brings that form of insulin resistance back down to normal. And well, I'm only showing you glucose tolerance data here, but we did insulin tolerance, we did insulin signaling, and corroborated these findings at those levels as well. Okay, but we're not the only ones that have ever worked in changing branched-chain amino acid supplementation, and the nutritional field is loaded with lots of prior work in this area. And if you round up the usual suspects, what you can find is you can make a list of studies that agree with what I've just told you, with branched-chain amino acids playing a role in promoting insulin resistance. And I won't go through all these, but you can see this. Some of these studies are in humans, some are in cells, and interestingly, the opposite manipulation of depriving diets of leucine results in improved hepatic insulin sensitivity, also seeming to involve the mTOR pathway. So there are many pieces of data that support the idea of BCAA playing a participatory role, but then there are a number of studies as well that say that branched-chain amino acids can actually serve a good function with regard to insulin sensitivity. So Zhang et al. At Columbia have found that giving leucine in the drinking water improves glucose tolerance. Chris Lynch at Penn State finds that leucine supplementation of drinking water is actually neutral with regard to features of the metabolic syndrome. And I'm aware of work from Mary Elizabeth Patti and Ron Kahn, who also have been giving leucine in mouse feeding studies and finding that it improves glucose homeostasis. Now, I will say that one common feature of all these studies is that leucine alone is given in the drinking water, okay? And it turns out that what leucine alone supplementation does is it lowers the levels of the other two branched-chain amino acids, valine and isoleucine. Whereas in humans, what we find is that all three branched-chain amino acids are elevated. So I don't think that these leucine alone supplementation models are a particularly good model of what we see in human obese and insulin-resistant states. And we gave all three amino acids in the diet and raised the amino acid levels in the blood to a level comparable to what we see in human obesity and type 2 diabetes. And so that's what I think is a potential validating point of our studies to this point, but more needs to be done in this area. Meanwhile, can we gain any further, let's say, confidence from following up in humans with regard to the relationship of branched-chain amino acids and insulin-resistant states? And so we've gone out in many different circumstances, and I'll be brief here. So Bill Krause at Duke recruits dyslipidemic and sedentary subjects in his STRIDE study. These span a wide array of BMI. So rather than a case control obese versus lean study, this allowed us to do a cross-sectional analysis with our metabolomic tools. And the other thing that Bill and Kim Huffman, his associate, did in this trial was to do the frequently sampled glucose tolerance test as the measure of insulin sensitivity. This is more rigorous and more precise than a simple HOMA score. And what we found in this population in a cross-sectional design is that once again the branched-chain amino acids and the related cluster of metabolites that I mentioned earlier were the principal component most strongly related to insulin sensitivity in this cohort, now measured as a frequently sampled glucose tolerance test derived calculated factor, or called the insulin sensitivity factor, or SI. Now what about bariatric surgery? Everybody's interested in bariatric surgery because it's the one thing that works for our obesity epidemic. And what we also know about bariatric surgery, intriguingly, is that when you look at subjects on bariatric surgery, they experience profound improvements in glucose homeostasis that seem to be far more than you would expect from the amount of weight loss. So we looked at two independent cohorts of bariatric surgery, one with Blandine Laferrere at Columbia, and one again with Laura Svetky in our home institution. And the design of these studies, both at the Duke site and at the Columbia site that Blandine heads up, was that we did gastric bypass surgery, this is Roux-en-Y bypass, and we also did a dietary intervention with the two groups being matched for amount of weight lost. Okay, so we're comparing gastric bypass to diet intervention, same amount of weight lost in two independent cohorts, one brought together at Columbia and one at Duke. And what we're looking at here in gray versus white is after the intervention and before the intervention. And what we're looking here at is a molar sum of the three branched-chain amino acids. And what you can see is that gastric bypass surgery lowers the branched-chain amino acids much more effectively than diet intervention, even though these groups of subjects lost the same amount of weight. And what we were able to show as well is that a principal component comprised of the branched-chain amino acids and related metabolites in this cohort was strongly related to the improvement in HOMA, whereas in the dietary cohort, this was not the case. So, moving on then to a cross-sectional study of another population. Many of you may know that Duke has started a medical school in Singapore, and through that I've had the good fortune to meet a wonderful epidemiologist in Singapore, Yishang Tai, who has his finger on every clinical sample in Singapore, I'm convinced. And I was able to say to Yishang, can we find 300 Chinese men and a similar number of Asian Indian men, all with a BMI of around 24. The purpose being we wanted to try to see if we could delineate a metabolomic profile of insulin resistance, keeping the BMI basically out of the equation. And it turns out that he was able to get these samples on which he had a HOMA assessment, and we could separate both the Chinese and Asian Indian subjects into an upper and lower quartile of insulin sensitivity, and then do metabolomic profiling on all those samples. And once again, the profile that clearly discriminates the insulin resistance is the branched-chain amino acids and related metabolites in now cohorts of completely different ethnicity, and with body weight taken out of the equation. Last but not least, this issue of can these metabolic profiles be prognostic for an intervention outcome? And now we turn to the multi-center weight loss maintenance trial, and with the Duke collaborators here being once again Laura Svetky and someone that many of you have heard about today, Dr. Svati Shah, and the WLM consortium. And what we did here is that there's several thousand subjects that have been characterized in this WLM study. We did studies on 500 of these, where we obtained a blood sample at baseline as these obese subjects came into these various centers to undergo a dietary behavioral weight loss intervention, the diet being the DASH diet, which is something that Laura is very involved in. And what you can see, this is the start point here, and this is HOMA at start. Each symbol is an individual subject, and if the symbol is down relative to the arrow I'm pointing to, that meant that the subject experienced a drop in HOMA, that is to say, an improvement in insulin sensitivity. If it's above, that means insulin sensitivity actually got worse. And here on the x-axis, you see the amount of weight lost over the 6-month dietary behavioral weight loss intervention period. And the thing that'll strike you immediately is that, unfortunately, the amount of weight lost during this intervention has essentially no predictive value whatsoever with regard to the improvement in HOMA score as a measure of insulin sensitivity. So here, if you look in this region of around 20 pounds of weight lost, you see some subjects with no improvement in HOMA and another subject that got a very nice improvement in HOMA, but there's no linear correlation whatsoever. So the question becomes, does a metabolic profile at baseline allow you to predict which subjects are going to improve with regard to insulin sensitivity as a consequence of the weight loss intervention? And so analyzing metabolites now only at baseline and performing principal components analysis, and then correlating that principal component factor score with outcome in terms of HOMA. Here are our typical principal components that we see in these kinds of data sets. I mentioned earlier long-chain fatty acids and ketone metabolites, clusters of medium-chain acyl carnitines, and here's our branched-chain amino acid related factor. And if you look at which, if any of these factors are associated with improvement in insulin sensitivity when measured at baseline, there's only one champion, and that is the branched-chain amino acids. And if you want to look at this data in a little bit more detail, this would be the upper quartile for the branched-chain amino acid factor score, the lower quartile for that factor score, and this is the change in HOMA. So if you have a high factor score at baseline, you have a good chance of improving in HOMA during the intervention, and you can see these p-values are quite impressive. If you have a low factor score at baseline, you're out of luck, so to speak, with regard to improvement in HOMA. And here's the box and whiskers plots that illustrate the same thing. Upper quartile, good improvement in HOMA, lower quartile, not much happening at all. So early days here, but the beginnings of a light shining that these kinds of metabolomic profiles can potentially be used as baseline predictors of intervention outcomes. And we're very excited about that. I want to continue to expand upon that. Now, let me finish the branched-chain amino acid story by coming back to, I don't feel like I've proven to you in any way that there's an absolute cause and effect relationship between branched-chain amino acids and insulin resistance. We've done animal studies. I've told you about them. The correlations are striking in humans, but we're not there at endgame yet. But now comes a very interesting paper that has just come out in the American Journal of Physiology by a young investigator named Dodie Sears, who trained with Jerry Oleski at UC San Diego. And what Dodie has been doing is examining thiazolidine diones, 4 different ones, which have different efficacy for normalization of glucose homeostasis in animal models of diabetes. This particular study was done in ZDF, Zucker diabetic fatty rats. And she did microarray analysis in multiple tissues as a function of treatment with these different thiazolidine diones with different efficacy for blood glucose normalization. And what she found is that far and away the most associated gene expression module among all those that she studied were this panel of branched-chain amino acid metabolizing genes. And she's taken here 2 genes from the branched-chain amino acid pathway and shown you the remarkable collinearity of the efficacy of these various thiazolidine diones going in parallel with their efficacy to change the expression of enzymes in the branched-chain amino acid catabolic pathway. So we have associations in the clinic, we can demonstrate some direct effects in animal models, and now when we look at therapeutic efficacy of at least this class of molecules, we see an association as well. So there's enough here where I think we have to keep driving this and really come to an understanding. And in a second, I'm going to tell you about how we think we're going to do that, because it's going to come, I think, in part from integration of metabolomics with other omic sciences, genomics and transcriptomics, to identify novel regulatory pathways. So we've been working both in mice and humans, and let me tell you about a very exciting collaboration that we've had with Alan Addy. Many of you know Alan. He's both a geneticist and a biochemist at University of Wisconsin, a brilliant and extremely energetic guy. And Alan, some years ago, came up with mouse models, the B6 mouse and the BTBR mouse, common mouse strains, where he integrates the OB mutation, the leptin mutation, and made fat B6 mice and fat BTBR mice, as you can see. Now, the difference is that BTBR mice, by the time they become obese and 10 weeks of age, are ragingly hyperglycemic, whereas the B6 mice are normal glycemic at that particular age, even though they're obese. So what you're dealing with here, basically, is a relatively diabetes-resistant mouse strain versus a diabetes-susceptible strain. So whenever a geneticist sees that, of course, the next experiment is to do a cross. And Alan didn't fool around. He made a cross of B6 and BTBR and made 500 F2 animals, where he pulled 6 tissues, including pancreatic islets, and did microarray analysis. And he also did whole genome SNP analysis on those animals. And then he came to me and he said, "Chris, come on, let's do metabolomics." So I said, okay. And so far, we've done this only in liver. But one of the things that is very exciting about doing metabolomics in the way that we do it, in a targeted, quantitative fashion, is that the concentrations of metabolites can very definitely be used as quantitative trait loci in the context of genetic studies, such as the one that Alan performed. So let me direct your attention here to a cluster of amino acids in the middle of the graph, which are mapping on this heat map to a specific region of chromosome 8, suggesting that there may be a gene on chromosome 8 that globally controls the concentrations of a whole panel of amino acids. So right away, that's kind of interesting. But the other thing that we can do, given that in this experiment we have whole genome SNP analysis, transcriptomic analysis, and metabolite profiling, is that we can integrate, because both the metabolites and the mRNA levels are tethered to specific regions in the genome thanks to the whole genome SNP analysis, allowing us to look for correlation matrices and allowing us to potentially identify networks of gene transcript metabolite or gene metabolite transcript. This takes very high-level computational methodologies, which I did not develop, which were developed by Alan and his colleagues at the University of Wisconsin, and then a graduate student mentored jointly by myself and Alan at Duke and Wisconsin, took all of this data and these computational tools. Christine Ferrara is her name. She's now in her residency training at the University of Pennsylvania. And one day Christine came to us and she said, "Alan and Chris, I found a pathway by which the amino acid glutamate strongly regulates PepCK, the key gluconeogenic enzyme." Remember, we're doing all this work in liver here. And this is going to happen through a network that includes nodes of alanine glyoxylate aminotransferase and arginase 1. Now, I got to tell you, I do not savvy. I do not fully understand the computational methods, the in silico methods, that led to this, the identification of this network. So being a practical man, I told Christine, this is great, but now we have to go back into the cells, and you need to show me that this network exists. So Christine dutifully goes to hepatocytes. She treats with glutamine, which is converted, of course, rapidly to glutamate, and she shows a 35-fold induction of PepsiK transcript in the hepatocytes, okay, and this is a log scale, let me point out, and a very nice induction as well of the other genes in this predicted node, the other transcripts in this network, alanine glyoxylate aminotransferase and arginase 1. So it appears that this network, in fact, can exist. But what is even more tantalizing is that if we now go back to the animals from whom the F2s were made, that is the B6 obese and the BTBR obese, and we look at glutamate levels in the livers of these animals, and we look at PepsiK transcript. Let's start with glutamate and appreciate that in both the diabetes resistant and diabetes susceptible mouse strains, as they become obese, glutamate levels in the liver drop. You can see that here. Now, in the B6 mouse, which is diabetes resistant, that drop in glutamate is correlated to a drop in PepsiK. So in other words, this network that we've described, where glutamate is proposed to control PepsiK levels, seems to be functioning perfectly well in the diabetes resistant strain, because as glutamate levels drop, so do levels of the PepsiK transcript. But in the BTBR, where the glutamate levels drop appropriately in response to obesity, this now seems to be totally disconnected from control of PepsiK, and you can see PepsiK levels are actually going up. So we think not only is this network a valid metabolic regulatory network, but we think it may be one of the things that goes awry in the progression of obesity to diabetes in the BTBR strain, that network becomes broken. So, emboldened by this, and buoyed by Alan's relentless enthusiasm, we've decided to move to a more challenging mouse genetics experiment. And we've just received word that we're going to be funded for this as a planning grant, an R24 planning grant. And I want to put it out to you all, because there may be members of the audience here today that when you see what it is that we're planning, it may trigger something in people from NHLBI or other institutes here where you might want to partake in what we're about to create as a resource. The brainchild, this is the brainchild of Gary Churchill at the Jackson Labs in Maine, and it's called the Collaborative Cross. And what's going to happen here is that Gary is going to breed 8 different strains of mice, some of which are very familiar to us, like the Black 6, the 29 mice, and others which are wilder strains, not the typical domesticated strains that we use in our research. And these 8 strains are going to be crossed every which way to Sunday, and to a point where we get 300 new admixtures, genetic admixtures, as strains. And once we have these 300 products of crosses with the genetics now all jumbled up in these animals, then there will be 17 generations of inbreeding, such that you now have 300 strains where you have replicate animals that you can study in biology. The purpose here is that one of the problems with mouse models is that they do not truly resemble the genetic heterogeneity of outbred human populations. And this is an attempt to create something that is more genetically robust, that begins to look more like outbred humans. And so we'll now have 300 recombinant inbred strains, all inbred, so that we can do replicative work and prove biochemical differences. The further idea then is to bring this team of investigators together, and on the planning grant at the moment we have Alan, myself, Gary, and Henri Bronengraber, who are going to measure transcriptomics, metabolomics, and metabolic flux and lipid and diabetic traits. We've spoken to Jeff Gordon, who stands ready to jump in with us as we generate more funding for the project, and the same is true for Tony Vidal-Puig. And so the whole point here is, as we look at whole genome analysis, as has gone on for some time in the field of diabetes, and we're a bit disappointed by the fact that we haven't identified genes that are sort of the single explanation for the genetic proclivity for type 2 diabetes. And what we're hoping here is that by going deeper in terms of the phenotyping of these multiple mouse strains, that we'll be able to connect genetic variability more effectively with physiologic metabolic variability, and in this way be able to better understand gene, transcript, metabolite networks in these diverse strains. So we hope to define genetic architecture that ultimately can be used to help us understand integrated causal physiological progression towards disease. So my invitation to all of you today, given that we may actually launch this project in the not too distant future, if we create this resource with all of this kind of profiling done in it, some of you who are interested in cardiovascular variability or other kinds of outcome may be very interested in coming along and helping us to phenotype in ways that are not embodied on this particular slide. And that's an invitation. All right, well, speaking of cardiovascular disease, we have also applied metabolomics to cardiovascular disease in humans, and we've done that in two major studies. My colleague Bill Krause at Duke has the gene card cohort. These are families with high incidence of early onset coronary events. These, as I say, these are families usually with multiple individuals within each family having heart attacks or other cardiovascular events before the age of 50. And then we've also worked in a non-familial group of subjects, the CathGen biorepository, and these are subjects that come to Duke for diagnostic cardiac catheterization, and this is random off the street. These are not familial populations. So when we do metabolic profiling in these families, we find, just as I just showed you in the mouse models, that metabolite profiles are highly heritable within families. So this gives us some hope that we're going to be able to use this kind of metabolomics to track with genetic variability as we move down the pipe. And I'm going to show you how we're going to do that in just a moment. This first study gave us the evidence that our metabolite profiles are highly heritable. But what I want to focus on here just for a second is the non-familial studies in the people coming in for diagnostic cardiac catheterization. And we asked two questions in these groups. First of all, because the cardiac catheterization segregates these subjects into people that have confirmed coronary artery disease versus those that don't, we simply wanted to ask in initially 180 CID cases versus 180 race and sex matched controls that did not have CID, and then in a validation data set of a separate 140 cases versus controls, does our metabolic profile pick out subjects with coronary artery disease versus those that don't have it? But what we then realized as we were beginning to assemble these coronary artery disease cases is that within these, there were many subjects who had gone on to have cardiovascular events during follow-up, myocardial infarction or death. So we were able to create then another case control comparison of subjects who, after cardiac catheterization, went on and had myocardial events versus subjects that did not have myocardial events. And we created then again a validation cohort, that's my stress earlier of how important this is, of 63 with death MI within 2 years and 66 matched event-free controls. So sort of two nested case control studies, one coronary artery disease, the other events, and both have an index and a validation cohort with them, okay? So we go in and we look with our targeted metabolic profiling, and the first question is, are there clusters of metabolites that associate with coronary artery disease in both the initial and the expansion or validation data set? And once again, the branched chain amino acids clustering here with the aromatics and the C5 acylcarnitines, just like in our original obese versus lean study, are correlated in both the index and validation data sets, and this association survives adjustment for typical factors such as obesity, smoking history, etc. And we've also found a urea cycle-related cluster that seems also in both the initial and validation data sets to associate with coronary artery disease. But the more interesting outcome, I have to say, was another cluster of metabolites comprised of short to medium chain dicarboxylated acylcarnitines, which did not associate with coronary artery disease, that's what I'm showing here, but rather associated with the cardiovascular events. So if you're in the upper quartile for this factor, here is your survival probability in this particular follow-up study. And if you're in the lower quartile, you can see, so very good separation here in terms of being predictive of subsequent cardiovascular events. And this survives adjustment for every demographic and medical factor that we can think of as well. This is a completely adjusted model, and you can still see that this factor 8, this cluster of small to medium chain dicarboxylated acylcarnitines, picks out the people that are about to have heart attacks. So we think that's pretty exciting, and we need to understand this better. So this is, again, an example of, here's a biomarker profile that's very exciting to my clinical collaborators, but as a basic scientist, I want to try to understand it better. So how are we going to do that? The two strategies that we're going to take are, first of all, we're now in a position to integrate metabolomics, genomics, and transcriptomics in the human population, because we now have profiled 3,500 CAF-gen subjects, 70% have CAD, 15% with type 2 diabetes. All 3,500 have targeted and quantitative metabolomic profiling. All have got GWAS, genotyping completed, and 1,800 will have peripheral blood gene expression profiling in the next few months by Illumina microarray. And we believe that this will allow us to better understand the genetic architecture underlying metabolic variability, for example, in the branched-chain amino acid related metabolites and the dicarboxylated acylcarnitines that seem to be a signature of imminent heart attack. What is the underlying genetics that drives those changes? And we think that that's an exciting prospect. Last but not least, what does the appearance of these dicarboxylated acylcarnitines mean? How do you produce these? And it turns out there's more than one pathway by which this can occur. One of those is omega oxidation of fatty acids, which occurs at the microsomal membrane, where you carboxylate, for example, this long-chain fatty acid at 2 positions to generate a dicarboxyl long-chain fatty acyl-CoA. And it turns out that these molecules, these dicarboxylated long-chain fats, are preferentially oxidized in peroxisomes, not in the mitochondria. So one of the things that these metabolites could indicate are changes in peroxisomal metabolism of fatty acids. But again, this is not the only mechanism that you can generate these by. You can also do this by a mitochondrial oxidation of other kinds of fuels, such as, believe it or not, branched-chain amino acids and other mitochondrial substrates. So our goal and purpose now, as basic scientists, is to understand which pathways generate these dicarboxylated species, which substrates influence that generation, and which pathophysiological factors, hypoxia, inflammatory stress, may also influence those pathways. So we can begin to understand how their appearance may relate to imminent cardiovascular events. Now, I thought I was going to be able to do cellular signaling mechanisms. I'll just show you the pictures. They're really nice because I'm out of time. I'm so sorry about that, because actually I have a really exciting story to tell here at the level of the mechanism of glucose-stimulated insulin secretion. This is how it works. I'm sorry. I never budget time correctly. I'm going to finish and just talk briefly about what I view as the future. So we're going to continue because we really believe in the targeted quantitative assay modules, we're going to keep building those. And it's a pain in the butt. You have to acquire or synthesize the appropriate stable isotope labeled internal standards. You have to develop extraction methods that work over a whole panel of chemically related analytes. You have to demonstrate reproducibility. It's hard work. It's analytical biochemistry. But you can see, I hope, that having these targeted methods can really aid in the game of clinical epidemiology and relating back to the other omics sciences. We're going to expand our use of the exploratory non-targeted metabolomics because I think we're really beginning to have confidence, particularly in this methodology where we have the retention time locked spectral library riding with us so that we can quickly move to targeted assays when we see things of interest. We're going to continue to integrate metabolomics with other omics sciences for identification of novel regulatory pathways, the collaborative cross being one example, and the big clinical cohort of cath-gen subjects being a second example that are on the imminent horizon. And I got to say that, you know, we live in very difficult times. I had a chance to speak with all my friends in the extramural group this morning, Maren Laughlin, Art Castle, others, Ron Margolis, and this is really difficult times for funding, and we all know that. And frankly, the practical aspect of this is having developed some of these tools in the academic arena, can these be used to better help us to fashion productive collaborations with pharmaceutical industry that will help us alleviate a period of some pain that is coming with regard to NIH funding. And I think that's something I want to have a very open dialogue with, because we're not trying to circumvent NIH funding. We're trying to find a way to do this at a time when it's going to be very difficult for NIH to do all the things that I know the good people here want to do. So let me stop there. Members of my academic laboratory I've tried to name as I went along that could contribute to the work. A big shout out to the outstanding scientists in our metabolic profiling lab. James Bain and Bob Stevens are at the faculty level. Brett Wenner, Olga Ilkayeva, Mike Muehlbauer are all PhD level scientists. And we owe a debt of gratitude in the early days at Duke to Dr. David Millington, who some of you know has run an inborn errors of metabolism screening lab using mass spec at Duke for many years. And he helped us get started with our own independent, science-focused endeavor. Shawn Burgess, Dean Sherry at UT Southwestern Medical Center, great collaborators over many years. Alan Addy and Mark Keller at the University of Wisconsin, and then outstanding clinical collaborators that I hope I've been careful to mention as I went along. So thank you very much for your attention.
I'd like to thank Dr. Newgard for his exciting presentation. We have time for one or two questions, and then we'll can take additional questions out in the atrium outside afterwards at the reception. Does anybody have a burning question? Come on.
So, Chris, that was a metabolic tour de force. What you showed, as I was trying to follow, was that some of these lipid profiles seem to predict the onset of cardiovascular disease. Have you been able to link that back to diet to say, you know, these particular people had a certain source of fats in their diet, and these people didn't?
Right. First of all, let me caution again that these metabolites don't necessarily come from fat. They can come from other sources, and that's one of the things we have to find out. And unfortunately, the cath-gen cohort, these are subjects coming in with suspicion of cardiovascular disease for a catheterization. We do not have dietary history on these subjects when they come in for that procedure. We do have a medical history, we have some medications history, but we don't know what they were eating on their way to that clinical encounter. Unfortunately. But yeah, it's a very interesting question, no doubt.
Hello. As scientists and being a part of this science community, we oftentimes we fail in our science, and you know, we follow the scientific method and we don't see the results that we want. But we still learn something through failure. We learn either what not to do, or we learn that we need to change our methods. And my question for you is, you talked about the metabolic profiling for these mice that you're going to crossbreed. and we know what we'll gain when you succeed, but what will you gain from it if you fail from this crossbreeding?
Failure is not an option. No, I don't mean to be cavalier. I think it's a very complicated project, and we may learn that we think that we can track metabolic variation to genetic variation, but when we actually go to test if a particular genetic variation truly leads to the metabolic phenotype that we're surveying in these mouse models, if we specifically go make that mutation and nothing happens, well, then we've learned that it's even more complicated than the most complicated experiment Alan Addy can think of. So that worries me, actually, if that's the case. But
Bert Gold, NCI. My stupid, and perhaps I just misplaced my biochemical genetic understanding. Is there anything bad about overfeeding branched chain amino acids? I mean, I know about maple syrup urine disease, but that's,
Right. People who are endurance athletes often supplement their diets with amino acids, including branched chain amino acids. People on the Atkins diet are dropping the carbohydrate content of their diet down dramatically, but leaving the protein and fat in place, seemingly similar to what I just described as a means of getting insulin resistance by virtue of adding branched chain amino acids. Here's what I think is critical. You have to be abusing all three macronutrient food groups. Too much carb, too much protein, too much fat. Where do we do that? We do that every time we go to McDonald's, right? And people, of course, fat and glucose have gained the notoriety. But what we're saying is, under the conditions of rampant abuse of all the food groups, is it possible that branched chain amino acids can play a role that has been heretofore unappreciated? Because it sure as heck maps with every change in insulin sensitivity that we can find in the human population. All right, thanks a lot.
Just a reminder, there's a reception out in the atrium out past the elevator. Please join us there. Thank you.